Welcome to another video in this series where we review the bio-decontamination process using VHP in GMP manufacturing environments. Let us continue to get an expert perspective from our subject matter expert, James Drinkwater, who is head of GMP compliance at Variances. James, how can we manage biological indicator BI quality variances, including rogue BIs, and make cycle development more efficient?
Essentially, the industry, pharmaceutical industry, has grown in the demand for isolators, driven by the revision of Annex 1. With that demand of growth of isolators and the essential qualification of the VHP cycle, then we need and the requirement for more and more BIs. With that increased demand in BIs, we've seen quality variances. We've seen an increase on rogue BI incident rates and also inconsistencies in those incident rates. So we need strategies to manage this very difficult to manufacture biological system, the BI. And they have now been developed and published through the PHSS as guidances how to manage these quality variances and particular rogue BIs and the impact on cycle development and qualification. Statistical analysis has also been applied to understand the significant impact on PQ qualification studies if rogue BI incident rates are very high. So as a quality control starting point, a strategy of pre-use qualification studies is required to mitigate impactful use of poor quality BIs, particularly the high BI rogue incident rates. At Variances, within our microbiological laboratory, we follow this published guidance and combine a number of studies in the QC pre-use qualification. So we take into qualification BIs that we know are already as best quality as they can be with the inherent variabilities. So these pre-use in qualification studies include independent d-value determination, population counts enumeration, scanning electron microscopy, which is an insight to the spore distribution with these SEMs, and a rogue incident rate study, which characterizes in a sample lot the incident of rogue BIs in that particular lot. And it's together, all those pre-use studies give us the position of starting with a good lot of BIs and less impactful on qualification cycle developments. And that is such an important starting point.
Thank you, James. Can you explain more about BI rogue investigation?
Yes, so if you use triplicate BIs in any location, that supports really the statistical approach, which may be one that you're justifying positive BIs by statistics. Not all regulators or companies are comfortable with having a justification of positive BIs in their cycle qualifications. We at Variances and many encourage to have just duplicates. So reduce the number of BIs. One of two would be an unexpected positive. So initially you have a growth and a no growth. The no growth is evidence that you have kill, the 6-log reduction. The no growth unexpected would be subject to an investigation. The qualifier for that is that at that location, you haven't had repeated BI positives, which means more of a process issue. So the consequence there is that at that location, we put triplicates because the chance of actually putting another rogue BI against the one that was originally there is very low. Consequently, we would expect those triplicates in an investigation cycle to be totally inactivated. And 10 times out of 10, we found if it's a rogue BI, they are inactivated, which means we have data to support and not just an assumption that it was a rogue BI. We also start to use in new technologies of enzymatic indicators. So here we can use one BI and an EI next to one another, and the EI gives us that interpretation if the one single BI was an unexpected positive. It gives us the interpretation it may be a rogue, then we go into the investigation as well. And it's together those approaches, those strategies enable us to sort of get robust cycle development that meets the expectations of regulators, the efficacy requirements, qualifications, and very efficient cycle development will help many repeat cycles because of the impact of poor quality BIs. So strategies are needed, strategies are now published. Follow them, we follow them, we encourage all to follow these strategies. Thank you.
Thank you, James, for clarifying those strategies for handling BI quality variances. As we come to the end of this video series, looking back across all the topics we have covered, what would you say are the key takeaways?
Yeah, for me, the key takeaways come from the fact that VHP biodecontamination process has significant strengths, but there are also limitations to manage and mitigate. That mitigation comes through process and scientific understanding. And with that understanding, we can actually manage the process variables under the banner of quality risk management within GMP requirements. We also need to make sure that that knowledge is in companies. So companies need their own training from a subject matter expert to make sure they have in-house knowledge so they can properly apply the mitigation. Another key point is that the vapor phase biodecontamination process is not comparable with dry fog. We've talked about the difference in the molecule and the sort of microns, say, droplets. And that needs to be understood. Without being comparable, you can't leverage data from the vapor published articles on dry fog. So it needs independent study. They're not comparable. And also the final point there is on biocompatibility. Even low VHP residuals around 1 ppm can impact proteins and biological products. So analytical studies now available, and we've characterized these from the lead from Genentech and used them in the Franzil chemistry lab to characterize the endpoint of cycles which are compatible with the process and not just the operator exposure level of 1 ppm. Typically 0.1 to 0.3 ppm is the endpoint today. VHP rapid decon hatches and rapid decon stations, which are standalone systems that support transfer of materials either into clean rooms or into the grade A space of isolators. These technologies have been developed to really mitigate the risk of manual disinfection of these very difficult to disinfect sterile packaged materials. Technologies now available as a solution. Another key point is process validity. This requires verification by what Annex 1 now defines as process monitoring, really to look at data that validates the cycle that you've just run is comparable with the original cycle you developed and qualified with biological indicators. So at Franzil, through the design space and measuring point concept, we apply process monitoring. And that is so important to support Annex 1 compliance, process monitoring, and support validation. Optimized cycle times are based on technical integration and features that facilitate initially the rapid rise to process lethal conditions, effective gas distribution, managing the condensable vapor, and accelerated aeration technologies using catalysts. Franzil employ all those facilities and techniques in our isolator designs, and we can support that for technical optimization. Another key point is managing BI quality variances, including rogue BIs, unexpected positives. Strategies based on QC studies to pre-qualify the BIs so the impact is mitigated are essential. In our microbiological laboratory in Franzil, we employ pre-qualification studies. We follow the published PHSS guidance on managing BI quality variances and rogue BIs. And we support this requirement, and we encourage all to follow this guidance.
Thank you to our expert, and thank you for joining us. Don't forget you can follow Franzil on LinkedIn for the latest news and insights.